Okay, so today we are going to move on and talk about how immune memory, that is the part of your adaptive immune system, that once you've successfully identified B cells and T cells that can help you defeat a particular novel pathogen, most likely resulting in your body producing lots of high quality antibodies to defeat that pathogen, how your ability to remember that pathogen through having memory B cells and memory T cells plays a very important role in your sustained immunity against that particular pathogen, potentially for your lifetime. And we'll also talk about how this concept of immune memory is a critical sort of foundation for the creation of vaccines. And then lastly, we'll talk a little bit about COVID-19 and then continue this discussion in the next lecture. So memory is a critical part of our adaptive immunity because without your adaptive system having immune memory, then anytime you are reinfected with that same pathogen that made you very sick, you'd have to start all over again with that process of selecting for lymphocytes that can help you recognize and defeat that pathogen. The very first time you are exposed to something that makes you sick, and when I say makes you sick, I mean some sort of novel pathogen that is something that your innate immune system can't deal with, we call that a primary response. In other words, the first time you have to wake up your adaptive immune system in order to come up with a effective defense. That's your primary response. Let's look at this here. So imagine that here comes googly-eyed drooling virus. It's something that your innate system can't deal with. So if you look at this figure, from the moment of first infection, we're going to assume that your innate response from your innate effector proteins and cells is not effective in eliminating this virus. So after several days, you begin to get very sick. Part of this is the inflammatory response of your immune system, causing things like fever, causing things like fatigue. So at a certain point, you have to wake up your adaptive immune system. So that's what we see here with these curves. The dark blue curve represents the levels or your population of T cells that have been chosen based upon the fact that their T cell receptors recognize certain peptides that come from googly-eyed drooly virus, right? So once those T cells are chosen, they multiply, and you have a big peak of these T cell populations. In the background, in light blue, is the antibody titer. In other words, at the same time, some of these helper T cells are helping to select B cells that have the correct shape, immunoglobulins, that can recognize antigen from the virus. And those B cells are selected, they multiply, some of them turn into plasma cells and produce antibody. So notice the time in weeks here. Over the course of the first week of infection, you get a large peak in antibody production. Remember, this peak represents wave after wave, right, of B cells turning into plasma cells, some of those B cells undergoing hypermutation to tweak the shape of their IG to come up with slightly better IG shapes. And those B cells then represent the next wave of plasma cells that send out antibodies. So even though typically from a viral infection, we may feel better by the end of 1 week, the reality is in the background, our adaptive immune system is still going strong, still selecting for T cells, and importantly, still selecting for affinity maturation of immunoglobulin shapes. In other words, still selecting for B cells undergoing hypermutation. So you notice that this high antibody titer basically begins to go down after 2 weeks. Right? So you've already felt a lot better. Maybe you went back to school, went back to work, but your immune system was still busy for another week, making high quality antibodies. And that antibody titer slowly goes down over several weeks because we evolved to assume that if you were exposed to this googly-eyed drooling virus in whatever environment made you sick, then chances are it's still hanging out in that same environment. You go back to school, you go back to work, and you're exposed repeatedly to this virus again. So these arrows here are repeated exposure to pathogen. But in each one of these cases, now you're talking about maybe a few months out being repeatedly exposed to it, your high elevated antibody titer flowing through your blood very quickly neutralizes any new infections, right? And that's an evolutionary adaptation to essentially protect us through the remainder of, the flu season, for example, or whatever season this googly-eyed virus, right, is kind of going through the population. So now what happens is after that, you have immune memory. Notice the light blue antibody titer now has dropped very low compared to your primary response, but it's still there compared to way at the beginning when you were first infected and you didn't have these high quality antibodies at all. So that elevated baseline level of this antibody may persist for months or even years. It takes the form of elevated levels of antibody flowing through your blood or in your mucous membranes, and that population of B cells that were the last B cells and the last plasma cells that successfully were producing antibodies to beat the infection previously. So that means after potentially a couple of years, you may experience a second infection. Right? So now, maybe 3 years later, you're exposed again. Right? This time, look at what happens. Your memory B cells, right, are already primed to respond to the site of the antigen again. So very quickly, these B cells turn into plasma cells and produce that same high quality antibody in tremendous amounts. So that's why, notice, your secondary response, right? We'll call it our secondary response because it's the second time you were exposed to this pathogen, right, after many, many years of immune memory. But the reality is, any subsequent time you're exposed to this down the line, you can also call it a secondary response. Right? So you create tremendous amounts of that high quality antibody, much more than in your primary response months or years ago. Part of the reason for this is that your immune system never really knows what memory B cells are worth hanging on to and what memory B cells may never be used again. After all, what if for the entire rest of your life you never were exposed to this same googly-eyed virus again? Right? Your immune system might just decide at a certain point, well, I don't think we're ever going to see this again. It's been years, so maybe let's forget about it. But if you do see it again, in this case, in the figure here, your immune system says, hey, this is clearly something that we are going to be re-exposed to, so we should really invest a lot of effort into a strong antibody response that's even stronger than last time. And that's why your secondary response is so high. This means that the combination of the elevated basal level of antibody in your system maybe was enough to neutralize the pathogen, but maybe the pathogen still had some sort of effect, still had some sort of infection response. And so with the big secondary response that you had, maybe you don't even feel sick, or at least the duration of your illness is much milder and shorter. So this is the important concept here, that when you survive a prior exposure to googly-eyed drooly virus, right, that is the source of your secondary immunity that will protect you from now on. So imagine in a primary infection, the first time you faced him here, this aggressive form of the pathogen made you very sick. So you were successful, though, in choosing T cells that can remember the peptides that the T cell receptor is the right shape for, that happen to be peptides that come from the virus. And you have B cells, right? B cells that had the right shaped Igs to ultimately make high quality antibodies. So if you survive the infection, then these guys are sticking around as your immune memory. That means an exposure that happens months or years later, which is your secondary response, here he comes again. Ha, nice to meet you, right? But your immune memory, right, protects you. So you are essentially saying, no, we've met before. So therefore, the aggressive form of this pathogen does not make you sick again. So consider this. The T cells, the B cells, they are recognizing essentially small peptides or small antigens. Right? They are not necessarily recognizing the entire virus. Right? They're recognizing little features of the virus, and that's all that matters. So what if the primary infection that you experienced was not from the aggressive form of the virus, but maybe you got infected with a weaker form of the virus? We'll call him the lazy virus. Right? So maybe he has mutated in such a way that he does not multiply and infect that quickly. So as a result, maybe you didn't really get that sick, but importantly, your immune system still learned. You still chose T cells and B cells that recognize features of the googly-eyed virus. Now, years later, the aggressive form over here now infects you. And once again, he says, haha, nice to meet you. But as far as your immune system is concerned, no, we've met him before, right? You met him in the form of the weakened pathogen that never really made you sick, but gave your immune system a preview, everything it needed to know, so that when the aggressive form did infect you later, you already had high quality antibodies and selected for lots of memory T cells to protect you. So the aggressive form doesn't make you sick. So if you consider, this is the basis of vaccines, the concept behind how vaccines work. Vaccines date back actually several hundreds of years, but British physician Edward Jenner is sort of credited as being the father of modern vaccines. So in the late 1700s, Jenner had heard stories coming from the Far East in places like India, where when villages were exposed to some sort of a pathogen that made lots of people sick, life-threatening pathogens, there were stories circulating that oftentimes the village wise men, the shaman, right, would actually have people line up at his hut, right? And what he would do is he would actually take scabs from the lesions of villagers who had gotten sick but who had survived, and he would actually make little cuts on the skin of healthy individuals and sort of rub some of this scab tissue in there. And what happened was these villagers may or may not get mildly sick from it, but more importantly, when they were exposed to the full strength pathogen, they were immune. So Edward Jenner heard this story and he wanted to actually try it. So if you look here in this cartoon here, smallpox was a very deadly, very dangerous and serious public health threat in the late 1700s. Smallpox killed millions of people because smallpox is highly infectious, and our bodies do not do a good job of defending against it. So what Jenner did was he he considered a related disease, cowpox. Cowpox is a disease that cows get, and they end up with little lesions very similar to the smallpox lesions that humans get. So what Edward Jenner did was he took tissue from infected cows that had cowpox, and he decided to go ahead, and the very first patient he had was the son of his gardener. So he put a scrape in this boy's arm, and he rubbed some of the tissue that came from the infected cow into the boy's arm. And because this was cowpox, the boy did not get sick. But amazingly, the boy developed immunity against smallpox. Why? Because if you look at cowpox and smallpox, these viruses are very closely related. So they have certain antigens in common on their surface, as represented here. So when cowpox is introduced into the human body, right, it doesn't make us sick because it's geared up to actually work in cows, not people. But your immune system will choose B cells that make eventually antibodies that detect some of these antigens. For example, this red antibody that detects this red antigen, this blue antibody that detects this blue antigen. So now, if you are exposed to the actual human smallpox pathogen, maybe the red antibody doesn't work. It doesn't do anything because on the smallpox virus, there are no red antigens, but there are the blue antigens. So therefore, your blue antibodies can now defeat the smallpox, right? As well as any other antibodies that you've produced that are shared. For example, the green antibodies. Right? So because this came from cowpox, which is the vaccinia virus, right? From Latin, vacca means cow. This is the origin of the word vaccine, right? It comes from cow. Because in principle, and this is an important concept, only the target antigens, like the blue antigen and the green antigen, need to be presented to the immune system in order to develop adaptive immune memory against the entire pathogen. So how do we actually make vaccines these days? For many years, the popular way of making vaccines, especially against things like viruses, was to take virus and either weaken it or inactivate it. So killed viruses, I put killed in quotes here because after all, a virus is not really a living thing. But if you treat it with certain chemicals or by heating it, you basically eliminate its ability to function and ability to infect. Now, if you are able to preserve, though, the three-dimensional shape of antigens, then this can serve as something that you can inject into the body. The body can then see those antigens and learn from it without the threat of this killed virus actually making you sick. So there's no risk of infection, but the downside is a dead or killed virus isn't going to elicit or cause much of any kind of evidence that an infection is going on. And remember, our immune system relies heavily on there being evidence that there's some sort of infection going on in order to effectively activate lymphocytes. So inactivated viruses are no longer preferred. What about weakened or attenuated? So weakened viruses can be made in the lab. Let's look at this cartoon down at the bottom here. So here you have a virus that infects humans, and this is the virus that you want to develop a vaccine against. So in the laboratory, in dishes of cells where you're growing human cell lines, these cells can be infected very easily by this virus, and not surprisingly, the infected cells produce many, many more of the infected virus, right? Now, obviously, you don't want to inject the fully active virus into a person's body because, yeah, right, that will definitely show the immune system the antigens that you want it to learn from, But this is like, shooting yourself in the foot, because you're essentially infecting that person and they're going to get sick from the virus, right? There's no need to do that, because that's what Mother Nature expects in the first place, that you're just going to breathe it in or eat it or something and get sick, right? But look what you can do in the lab. If you took that human virus and you had a culture of monkey cells in blue here, monkey cells are close enough to human cells that maybe the virus can, in fact, infect these cells and to a certain extent, right, multiply. But they will multiply much slower in monkey cells because they're really geared up to work in human cells, and in monkey cells, there's enough differences that the virus is kind of slowed down. However, if you keep reinfecting monkey cells with the virus, you create a selective pressure on the virus to mutate, right? And eventually, you can see here, the virus will mutate in such a way that you end up having the virus altered so that it now can multiply very easily in the monkey cells. Right? And what that means is that these viruses now, which are represented as yellow, are now kind of geared up towards monkey cells. So now if you go ahead and introduce the human body to these altered viruses, right, they're not going to infect and multiply as quickly in human cells anymore, because you've altered them to be geared up for monkey cells. But at the same time, they are presenting antigens that you would like the human immune system to see, right? So attenuated viruses like this had been used for several decades, but they have some advantages and disadvantages. The advantages are they are quote unquote live, right? Because they can still infect cells, even though the infection is slower. So that yields a better immune response because your immune system actually does see that some of your cells are getting infected. So there's the release of cytokines that alerts your immune system, hey, wake up, start choosing B cells and T cells that can recognize this thing, right? But the downside is, just like these viruses were mutated in the lab to become weakened as far as human cells are concerned, in the human body, they might mutate again. And you might actually end up restoring their original potential to infect humans. So in reality, there are cases in history where attenuated viruses were used in vaccine formulations, and it caused problems. So the best example of that is probably polio. In the 1950s, tens of thousands of people in the US got sick from the polio virus. And you may have seen old, grainy black and white photos of hospital wards, of pediatric wards, where kids they were basically in wheelchairs. They were crippled because of the polio virus. So there was a real desire to come up with a polio vaccine. So Jonas Salk came up with the vaccine based upon an attenuated or weakened polio virus, and it was very effective. So then for the next couple decades, there was a very aggressive vaccination campaign where people were injected with this weakened form of the polio virus so they could develop immunity and then become resistant to the actual full-strength polio. But by the 1990s, this was so effective that polio was practically eradicated. In fact, the only cases of polio that would show up in the United States every year were maybe a dozen or so cases, but these cases were actually caused by the polio vaccine. The weakened virus in the vaccine, just by bad luck, randomly mutating again in the patient's body and becoming full-strength polio and giving that person polio. By the early 2000s, they reformulated the vaccine, so it's no longer based upon a attenuated virus. And now that's the polio vaccine that all of you probably got. Okay, so mutations allow viruses then to escape the immune response. Because again, mutations that alter the shape of certain antigens that are on the virus, that are the antigens that your immune system is recognizing as part of its defense and part of its memory, if those shapes change due to mutations, then the effectiveness of your antibodies can be eliminated, right? So the flu is a virus that mutates a lot. Flu spreads very quickly through populations of unprotected individuals. So when you have local outbreaks of the flu, we call that an epidemic. Right? The flu is an RNA virus, so it carries its genome as RNA, and it's very prone to mutations. Right? So you might actually have situations where you get the flu, develop immunity against it, right? But then in the process of when you were sick, pass the flu on to somebody like a friend, and in their body, the flu might mutate into a form that once you see your friend again, your immunity against the original form of the flu doesn't protect you against the one that your friend gives you. So let's look at this here. Let's look at the cartoon. So you're exposed to the flu. Here's the flu virus. A particular antigen that's very prominent on the virus is this hemagglutinin, which is indicated by this H in this shape, right? So this is an antigen that would be all over the surface of the virus capsule, and that is one that your immune system oftentimes uses to select antibodies against it. So your immune system selected B cells that make this blue antibody and this green antibody, and they neutralize the virus and block its ability for the hemagglutinin molecule to bind to the surface of your cells. Therefore, these antibodies block the virus from infecting. Right? So in the meantime, maybe one of your friends got infected by this virus also, and in their body, the virus mutates. So you see now the virus that they're producing every time they sneeze or cough, the shape of the hemagglutinin is a little bit different, right? That's what this sort of yellow part represents. Right? Ironically, now you go back and see each other in the classroom or at work, right? And your friend sneezes on you, your antibodies that you produced originally against the virus may not be effective anymore. For example, the blue antibody may still work, but the green one, right, that recognized this part of the hemagglutinin antigen no longer works because that part of the antigen has mutated. Right? So we call this antigenic drift, meaning it's the same virus, right, but the antigens have sort of changed and they're drifting away from the original set of antigens that were presented when this virus entered the population and started infecting. So every 10 to 50 years or so, this drift basically takes the virus far enough from its original form that it basically causes now a worldwide pandemic, right? Where populations that previously were immune or resistant to the virus, right, maybe a decade ago, right, now their immune memory isn't good enough to protect them against the new mutated form. So what that means, really in a practical sense, is every year when the Centers for Disease Control identifies the strains of flu virus that are breaking out all over the world, and they predict or anticipate that these are the new strains that are going to hit us the hardest, right? They develop new versions of the vaccine, right, so that it's updated to effectively show our immune systems the latest version of the different antigens that the new strains of the virus are displaying. So if you look at this here, imagine you are infected by the virus. Okay, this is a flu virus. And as a result, you know, the book is a little overly dramatic here. They like to have this, how many days in bed were you sick, right? So maybe the first time you're infected, it's really bad. You were laid up in bed for 14 days, right? And of course, that's your primary response going on in the background, right? The first time you're exposed to this. So as a result, let's say the flu at that time has these 4 antigens, A, B, C, and D, and you end up selecting for B cells in your body that know how to make antibodies against A, B, C, and D. All right. So now let's say some time passes, right? Maybe a few months, and you are reinfected with the virus, but the virus has mutated in the meantime, right? And so now it still has A, B, and C antigen, but instead of the D antigen, that antigen has changed enough that we'll call it E. Right? So as a result, only 3 out of 4 of your antibodies are effective against this. And as a result, maybe you get sick again, right? But you're sick for only a day, right? It's just a day of feeling a little funny, maybe, right? Not even enough for you to really, really feel like something's wrong. But then a few months later, right, the virus has mutated to the point where only 2 of your antibodies are relevant, right? Or it mutates so that only 1 of your antibodies still works. And maybe a whole year later, right, the version of the flu that's now going around, all 4 of those antibodies have changed enough that when you're re-exposed to this, you're going to be laid up in bed for 2 weeks again, because none of your antibodies that you developed last time work. And you have to now develop a brand new set of antibodies, right? So that means even though you got the flu shot last year, you still should get the flu shot this year, right? So that it can provide your immune system the updated preview of what this year's flu antigens look like. Right? So worldwide pandemics often are caused by recombinant viruses. These are viruses that are particularly dangerous because they developed or were harbored in some other kind of animal. And they're really geared up for that particular animal, right? So they may have certain features that our immune system, the human immune system, are just simply not familiar with. Right? The danger happens when different viruses from different animal reservoirs, I'm referring to different species of animals as reservoirs where different viruses are essentially building up. If they have the opportunity to mix and recombine their genetics, that's a recipe for disaster. Let's look at this. Let's say you have a flu virus that makes people sick, right? So this human virus is one that, number one, infects human cells very well, right? It spreads quickly in the cells. And number 2, right, has the potential to make you sick, right? You know, not necessarily deathly sick, but definitely sick in bed, feeling miserable with fever, maybe throwing up for a couple days at least, right? Let's say there's an avian flu, a bird flu. This bird flu, right, is the wrong shape to infect human cells. So even if you breathed it in, it doesn't work in human cells. But let's say this bird flu does infect pig cells. Now consider this scenario where this pig here breathed in both viruses, the human flu virus and the avian flu virus, at the same time. Now you might say, well, how likely is that? Well, consider that when we show cartoons of a cell that are being infected by a virus, for simplicity's sake, right, we often just think about it as being, oh, one type of virus infecting that cell. But why should it have to be limited to that? Right? If this pig breathes in both types of viruses, then both types of viruses, the human red ones and the avian blue ones, might be infecting the same pig cell. Inside the pig cell, so it makes the pig sick, but inside the pig cell, all the genetic material of these virus particles are mixing around. So maybe the new virus particles that are made have a combination, right? They have altered antigens now that determine what types of animal cells they can infect, and importantly, right, the mechanisms of infection inside the genetic material are very different. So now, if a person breathes this in, it may be a very bad combination of this recombined virus that has jumped from species to species might have mutated so they can really infect human cells. And the scary thing is, because genetically they're very different than your typical human virus, your immune system might not be very successful in defending, and you might get very, very sick as a result, because this is a strain that has never before been seen in humans before. Right? So not surprisingly, that's what SARS-CoV-2 is all about. That's what the COVID-19 pandemic is a result of. So these coronaviruses, like the SARS-CoV-2, right? I mean, they've been around for a long, long time, right? So coronaviruses are not something new. Throughout the history of recorded mankind, for example, all the way back to the 1100s, right, there were documentations of some sort of virus that spread and killed millions of people. Right? And we believe that that virus jumped from bats over to humans. Right? The original SARS in early 2000s, we believe, came from bats, went through some sort of intermediate mammal before entering humans. And then in 2012, there was MERS, right, that went from bats into camels into people. So the SARS-CoV-2 that originated, as best we can tell, from Wuhan in China, right? We're still not 100% sure what animal reservoir it jumped from. Early in the pandemic, there was thought that it came from bats and might have gone through some sort of mammal like a pangolin, right? It's like an armadillo-like animal into people. But I think at this point, they are still not sure what animal it really jumped from. But again, the concept is because it comes from a different species and is mutated in a different species, right? By bad luck chance, it mutated such that it can infect human cells, but it carries all kinds of weird things in it that human immune system is just not used to seeing because it doesn't look like a typical human virus. So here's SARS-CoV-2. They are called coronaviruses because when you look at them under high magnification, they look like crowns, right? They have these sort of like spikes around them on the surface. What these spikes are these protein spikes that are made of glycoproteins. These spikes actually are the shapes on the outside of the virus that match up with certain proteins on the surfaces of the human cell. So in the case of COVID-19, the protein spike is the right shape to match up with a particular receptor that's on the surface of human cells in the respiratory tract known as ACE2. And by sticking to the ACE2, that's how the virus ends up sticking to the surface of the human cell and essentially then being absorbed or entering the human cell to start infection. So here's a model of the surface of the virus particle. And here is the accurate three-dimensional shape that was generated very early in the pandemic. You know, as early as February 2020, researchers already determined this is the shape of the glycoprotein spike on the surface of the antigen, right? Because these spikes are on the outside of the virus, these are the types of antigenic signals that are ideal for using as vaccines, because antibodies that you ultimately want to produce are going to bind to the outside of the virus by matching up with these shapes. Right? So the concept is, can you introduce this shape into the human body so that the human body can use it as a preview? You will choose B cells that have the right shaped immunoglobulins that recognize this glycoprotein spike shape. Right? They will work with helper T cells that also recognize peptides that come from the glycoprotein. And together you can create plasma cells that will produce antibodies, right, so that the vaccinated individual, right, their immune system never would have actually seen the entire virus particle, just the spike protein. So that way there was never any risk of the patient really getting COVID from the vaccine. But hopefully that preview of the glycoprotein spike was enough so that the person's immune system was already now producing high quality, for example, IgAs in the mucosal lining of the respiratory tract. So when the real SARS-CoV-2 virus gets breathed in, it can be neutralized by the virus. Okay, so that's where we're going to stop today. When we continue with this discussion next time, we'll look in greater detail as to how these new vaccines from Moderna and Pfizer were actually produced to be able to successfully introduce this glycoprotein spike shape into the human body. All right. Okay, see you next time.